Welcome back to week 8 of Applied Immunology. We started off this week with an overview of the comprehensive immune response to infection, and we're going to build upon that today by learning how we can artificially induce this type of response in the absence of infection. And this is using an important preventative therapy called vaccination. The vaccine unit for Applied Immunology will be covered by today's Lecture 8B on general vaccine design and mechanisms of responses, as well as an optional Lecture 8C, which will specifically cover the various SARS-CoV-2 vaccines that have been approved in the last few months. I know that many of you are probably interested in learning more about these vaccines, but I've made Lecture 8C optional in case anyone here has dealt with personal loss or hardship as a direct result of the COVID-19 pandemic, in which case you might not want to spend an entire lecture talking about it. Today's Lecture 8B will cover more broad aspects of vaccine design. We will start off with a conceptual discussion of vaccine benefits and safety, then move into a more scientific overview of vaccine formulations, their mechanism of action, and factors that influence or impact vaccine efficacy. I'm excited to cover the topic of vaccines because this is just such an important translational application of what we know about the immune system. And despite this importance, there's an obvious need for increasing scientific literacy and understanding of vaccines within the general public. We last talked about vaccines way back in our introductory week 1 lectures, where I introduced the effect that immunization programs have had on communicable childhood diseases. These graphs represent data sets from the United Kingdom, where they show the incidence of various infections in children after the introduction of public health immunization campaigns. In recent years, it might be easy to lose sight of how incredible these decreases are, especially considering the terrible effects that some of these infections had. So polioviruses can infect and destroy peripheral nerves, leading to permanent paralysis in a portion of infected children. Meningococcus bacteria can cause a form of brain inflammation called meningitis, and this has a case fatality rate of 10 to 15% out of those who were infected. Measles is a very highly contagious paramyxovirus that doesn't have a high mortality rate, but about 30% of patients will develop severe complications like pneumonia, encephalitis, or corneal damage that leads to blindness. Overall, the World Health Organization estimates that anywhere from 2 to 3 million lives are saved every year by childhood immunization programs, and there's still certainly room to improve upon this by increasing vaccination efforts in less wealthy countries. The benefits of vaccines extend even beyond control of infection, and this is the case with human papillomavirus or HPV infection. So during infection, HPV expresses oncogenic proteins that lead to the development of invasive cervical cancer. This study from Sweden looked at the rate of cervical cancer development amongst young women. And you can see from this graph that HPV vaccination in women over 17 years old, shown by the blue curve, had slightly decreased cervical cancer incidence compared to unvaccinated women in the orange curve. Most impressively, women who had received the HPV vaccine before the age of 17 showed essentially a flat line in cervical cancer rates, as shown in the green curve. So not only does the HPV vaccine obviously prevent HPV infection, but as a result, it also prevents cancer development that's caused by that virus. These types of reductions in disease incidence are impressive when you think about the overall health and well-being of the general public. Like I said during week 1, I would argue that vaccines, together with antibiotics, represent two of the most significant developments in modern medicine. It's also important to consider the effects that vaccination has on the hospital and public health systems as a whole, since immunization essentially removes large cohorts of patients who may otherwise have been hospitalized either due to acute infection symptoms or long-term effects ranging from permanent disability to cancer. Preventative therapies such as vaccines basically relieve what would otherwise be a massive economic and infrastructural burden on both hospital and public health systems. So, given the enormous benefits that childhood immunizations provide in the context of public health, why is there still so much suspicion and mistrust about this type of preventative therapy? I think that this is a very complicated question that requires consideration of numerous social, cultural, and psychological factors, which we will touch on briefly. But it's easiest to start by bringing up this one paper by a physician named Andrew Wakefield that was published back in 1998. This was published in a peer-reviewed journal called The Lancet and examined children who developed autism symptoms within 1 month of receiving the measles, mumps, rubella, or MMR vaccine. The group proposed that there was a mechanistic link where MMR vaccination caused intestinal inflammation that then led to neurodevelopmental abnormalities. The authors referred to this as autistic enterocolitis and stated that this was a direct result of MMR vaccination. Now, this paper and its authors' motivations were riddled with issues that ranged from flawed study design and experimental readouts to more egregious ethical concerns related to data fabrication, patient violation of patient beneficence, as well as conflict of interest. So there were issues with using a tiny cohort of only 12 patients with no negative controls, and these patients were actively selected by Wakefield based on his knowledge of their medical history rather than being randomized. There were also some other problems with the actual experimental readouts being used as data to back up these claims. Beyond these errors in study design and data collection, it later came out that there were serious problems with data fabrication and the manipulation of data values, which obviously are a form of research fraud. Additionally, it turned out that Wakefield was being paid as an expert in a class action lawsuit against vaccine manufacturers. In addition to planning on marketing diagnostic tests and as well as alternative measles vaccines. So these are obvious forms of competing financial interests. I also want to highlight that this work was an extreme violation of a key medical ethics principle called beneficence, which states that clinicians must select treatments based on maximizing benefit and well-being to a patient. This group performed pretty invasive medical procedures on a group of autistic children for reasons other than properly researching their medical condition. This by itself is very sad and I think quite upsetting. And taken together with everything else, this paper represented truly egregious ethical violations and research fraud. It was eventually retracted by the journal, Wakefield's medical license was revoked, and he has since been completely discredited by scientific and medical communities. However, the long-term damage was done at this point, as this really represented the birth of the modern anti-vaccine movement that's gained a lot of momentum over the last couple of decades. This controversy ended up having a lot of power for a number of reasons, a major one being that it preyed on some very real fears of parents that they may have approved a medical procedure that could have actively harmed their children. And I think this highlights just how irresponsible this study was, since these doctors essentially took advantage of trust that was placed in them as members of the medical community. This has since blown up into an entire anti-vaccine movement with serious long-term consequences related to decreased vaccination rates, both for childhood diseases, as well as likely contributing to some of the vaccine hesitancy that we now see with the coronavirus vaccines. Since the Wakefield paper, there have been many studies that conclusively refute any link between the MMR vaccine and autism development. This table here summarizes some larger ones involving tens of thousands of patients and concluded that there's no correlation between these two things, never mind an actual mechanism that links the two. This holds true for other vaccines besides MMR as well, and these studies have continued throughout more recent years. This is a bit frustrating when you think about how many groups have invested their time and money into refuting a claim that vaccines cause autism, because these are all resources that would have been better used to study actual underlying causes of autism and colitis, but instead they've been redirected towards disproving this one claim whose reception by the public got blown completely out of control. Despite this body of evidence, the Wakefield paper has an unfortunate legacy of opening up all other sorts of concerns about vaccines besides autism. I think that this is at least partially due to the fact that it was published right around when the internet started playing a much more prominent role in people's lives, and this has allowed people to connect with fringe theories online much more easily than ever before. Besides autism, people have moved their focus to vaccine components that they consider unnatural, such as thimerosal. There's also the claim that immunizations are scheduled too closely together in a way that, quote-unquote, overwhelms the immune system, which is factually incorrect since the immune system is actually designed quite well to handle repeated exposures to inflammation. Other concerns range into brand new territory, like proposing that vaccines are part of a government plot to control the population through injection of microchips, and other fringe theories that I'm just not going to address today. The tricky thing about the anti-vaccine movement is that this is essentially a conspiracy theory that's grounded in an inherent opposition to these treatments. It's more of a philosophical opposition, and that this exists independently of any data related to the topic. Belief in these theories may come from a place of fear for someone who doesn't understand how vaccines work, and this can intersect with more complex topics like a mistrust of pharmaceutical companies, or opinions concerning personal liberties where people resist mandated forms of medical treatments. I also want to quickly make the distinction between the anti-vaccine movement, which I think is categorized more along lines of a conspiracy theory, versus the concept of vaccine hesitancy. So you may see vaccine hesitancy with people belonging to groups that have been historically mistreated by doctors or government studies, in which case they have a more valid mistrust in the medical establishment. So there are different underlying factors that drive the anti-vaccine movement versus vaccine hesitancy. Regardless of the exact reasoning people use to justify their stance on vaccines, it all comes down to this core belief that the risks of vaccination outweigh the risks of the diseases that those vaccines prevent. As we will learn in the next slide, this isn't the case for most diseases, but it can be difficult to convince someone otherwise, especially if their anti-vaccine sentiment is rooted in deeper philosophical beliefs rather than objective data. As students that understand immunology, our best course of action is to learn about the biological basis of vaccines, the actual risks associated with vaccines, and how these are balanced by their benefits, and then just do our best to communicate that information in a respectful way to non-scientists who may be either overtly against or just slightly hesitant about taking vaccines. If we learn about the actual evidence for side effects and risks of vaccines, it's impossible to argue that these are worse than the risks of natural infection with a lot of these pathogens. Before vaccines, communicable childhood diseases drove high rates of infant and child mortality in response to some infections, while other infections led to permanent disfigurement or disability. Vaccines are kind of a victim of their own success, in that as memory of these infections recede from the collective memory of the general public, people forget about what these infections actually used to do to children. And this makes it easier to take the actual side effects of vaccines and blow them out of proportion. Aside from public health harm, the recent backlash against vaccines also overshadows actual research that's being done to understand scientifically valid side effects of vaccines. This infographic sums up different types of vaccine side effects, ranging from more common ones at the top of the graphic versus rare or inconclusive effects at the bottom. Phase 1 trials of vaccines are extremely stringent with respect to their safety standards and monitoring patients, so they pick up on fairly common side effects like swelling, soreness, and mild fevers that I'm sure everyone here has experienced following a shot. There are also rarer instances of fever-induced convulsions, which can happen on the order of 1 in every 3,000 patients. However, trials usually aren't able to enroll large enough numbers of patients to catch even more rare side effects that happen on the order of 1 to every 10,000 patients or fewer. And these types of extremely rare side effects include some allergic reactions, certain types of bowel pathology, and a form of paralysis called Guillain-Barré syndrome that's so rare it's almost impossible to conduct conclusive studies on it as a side effect. Note that all of these side effects occur early on post-vaccination, and there are no known long-term side effects of vaccines. Researchers therefore need to collect extensive monitoring data in patient populations outside of clinical trials in order to get enough patients with rarer side effects to be able to study anything with a degree of statistical strength. The goal of these studies is to identify true side effects from vaccines, and this is done in order to learn how to better screen patients and identify those that may have severe adverse reactions to a vaccine. This type of research is essential for building and maintaining public trust with vaccines, and this is especially important given the amount of misinformation that is being circulated from all other sorts of non-scientific sources. The importance of this research is clear when we look at some of the risk factors that we know are associated with serious side effects. A major one of these are undiagnosed forms of immunodeficiencies in children who have compromised immune systems and may not be able to tolerate live attenuated vaccines. Another important consideration for children is whether they have allergic reactions to certain vaccine components, since this would put them at a higher risk of more severe complications like anaphylactic shock. And these types of things highlight why it's important to conduct research that helps us understand actual risks that are associated with vaccination. I know that the first parts of this talk have been centered on people's perception of vaccines, but I think that it's important to understand the broader social context surrounding these things before we get into the scientific details of how they work. Scientific and medical advances do not happen in a vacuum, and this is especially true for something like vaccines that have such a major impact on a field like public health, which is influenced by many factors beyond biology. Okay, that's enough of my ranting about public health for this lecture. Let's move on to learning about the biological mechanisms of vaccines. So vaccines can take many different forms, but at their heart, any vaccine is defined as a biological treatment that safely induces a protective immunological memory response against a given pathogen. And importantly, it has to do this without active infection by that pathogen. Given this definition, what are some of the shared features of effective vaccines? So first and foremost, vaccines must be safe and not cause illness or death themselves. Given the long discussion we just had about anti-vaccine concerns, I want to stress that safety is an absolute top priority of effective vaccines, and that these products are tested extensively in clinical trials to ensure that they are well tolerated. Vaccines are also often formulated with the goal of reducing side effects, even if these are minor ones like muscle soreness or fever. After safety, vaccines obviously have to be protective, so they must induce long-term immune memory responses that confer sustained protection from a pathogen. This is achieved by stimulating an adaptive immune memory response against specific antigens from that particular pathogen. As we will learn shortly, this antigen can be introduced using a number of different strategies, which we will learn about more in the upcoming slides. Lastly, effective vaccines should ideally address a number of practical or infrastructural concerns related to their cost of production, their stability and storage, as well as their ease of preparation and administration. These aspects of vaccines are extremely important with respect to being able to successfully carry out vaccination campaigns in areas that are not well served by more advanced hospitals and transportation systems. These non-biological considerations are extremely important with respect to health equity and accessibility. Now, there are several ways to design vaccines capable of inducing long-term immune memory against a specific pathogen, most of which differ in terms of how they introduce antigen to the immune system. The first vaccine that we will cover is typically seen as the most effective, and this is something called a live attenuated vaccine, which is primarily done with viruses. These vaccines consist of a live virus that is capable of initiating infection in host cells, but has an impaired ability to complete full infectious cycles or replication in an immunocompetent host. And this is something that we call an attenuated viral strain. This is usually the most effective type of vaccine since they elicit robust immune memory responses against numerous antigens found in an entire viral particle. And this can activate polyclonal immune responses. Attenuated viruses also express numerous viral PAMPs that can stimulate innate immunity through pattern recognition pathways. So they kind of strike a fine balance between causing enough viral entry and maybe even some low levels of replication to robustly stimulate immune responses, but these need to be attenuated enough that they don't actually cause a productive infection. The caveats to using live attenuated vaccines is that they can't be used in immunocompromised patients, since the attenuated strain may actually be able to function more like a fully virulent strain in these people. Another concern is the possibility that an attenuated strain could acquire mutations that allow it to revert back to a more virulent strain. The probability of this is very low, but it still remains a safety feature to keep in mind with attenuated vaccines. Lastly, another drawback of live attenuated vaccines is that in order to make them, you need to use something called a heterologous cell culture system, which just isn't available for all viruses. We make attenuated viral vaccines using something called heterologous cell culture. So this consists of taking a virus that is adapted to a human host. So at first, it's perfectly capable of infecting and replicating in human cells. And then you take this virus and culture it in vitro with cells from a closely related but separate species, such as monkey cells. And we define these as heterologous cells. The virus is passaged serially for many generations in these heterologous cells, with the goal of adapting the virus to the other species' host cells through mutations in the virus's genome. Once the virus is adapted to the other species' host cells through these mutations, it can no longer replicate in human cells, and so it's eliminated by the human immune system. And this is what makes it an attenuated strain. Although live attenuated vaccines are typically the best vaccines, you can't make attenuated versions of every target pathogen. Some viruses just won't infect other species' cells in heterologous cell culture, often because they have highly specific requirements in binding to entry receptors on a host cell's surface in order to initiate viral entry and infection. So in these cases, different forms of vaccines need to be used instead. All other types of vaccines fall under the umbrella of non-live vaccines, since they are not based on infection with a live pathogen. Because of this, they are generally seen as safer, although this does come with the trade-off that killed vaccines or non-live vaccines usually don't work quite as well as live attenuated vaccines. There are a variety of these in use, which differ with respect to how exactly they introduce the antigenic component of the vaccine, which is used to direct the specificity of immune memory. Some vaccines are made using killed whole organisms, where viruses or bacteria are treated with heat or chemicals that kills or inactivates them, and this renders them non-infectious. These may not work as well as attenuated vaccines, since some antigens or PAMPs may have been degraded during the killing process, so they don't exhibit as robust long-term immune memory responses some of the time. An interesting note with these vaccines is that in order to generate large quantities of some viruses, this protocol involves growing virus in fertilized chicken embryos inside of eggs, so trace amounts of egg proteins may be found following isolation of these viral particles, which is why some of these vaccines come with a warning to not use them in patients that have egg allergies. Other vaccines are based on administration of individual antigens that are associated with infection. Toxoid vaccines, such as diphtheria and tetanus, can be used against bacterial species whose disease symptoms result from production of bacterial toxins. These vaccines are made using inactivated forms of these toxins, and these actually elicit really nice neutralizing antibody responses that will protect immunized patients from toxin entry into host cells. Subunit vaccines contain purified proteins or polysaccharide antigens from pathogens, and viral-like particles function similarly to subunit vaccines, except that these protein subunits are attached to a synthetic particle that makes them sort of physically look more like an actual virion rather than just purified bits of protein. There are a couple of formulations like polysaccharide conjugates and vesicles that I won't get into today, but I want to briefly touch on the last two in these charts, since they're the types of antigenic components used in some of the current SARS-CoV-2 vaccines, which are viral vectored and nucleic acid vaccines. Rather than administering viral proteins directly through any of the above strategies, these nucleic acid vaccines instead operate by introducing a nucleic acid that encodes a viral protein antigen to host cells. And then the host cells are responsible for translating the RNA to produce the target antigen by the host cell. Vector vaccines do this by using a viral vector, which is usually something like an adenovirus, while nucleic acid vaccines do this by encasing a physical piece of RNA into a lipid shell that's called a liposome. We will discuss each of these more extensively in the optional lecture 8C for this week, if you're interested in learning more specifically about the SARS vaccines. These different types of vaccines work by using various strategies to introduce a pathogen-associated antigen to the body, and this is what is targeted by specific immune memory responses. But aside from the antigenic component, or the antigen payload of vaccines, let's learn about some of the other things that go into a vaccine dose. One of the other most important biologically relevant ingredients in vaccines is something called an adjuvant. Adjuvants are small molecules that may be included in a vaccine to help boost innate immune responses to the antigen payload. Adjuvants are important for non-live vaccines, where they stimulate innate immune cells to respond to inflammatory cues that are introduced alongside antigen. There are not a lot of different adjuvants that are approved for human usage, but some common ones are forms of aluminum salts or lipid-based emulsions. Adjuvants are very safe compounds to use, since their concentrations in vaccines are less than what you would find in things like drinking water or several types of oral medications. Vaccines also need preservatives to help prevent contamination by bacteria or fungi. And this is a particular concern if you have one vial of vaccine stock that's repeatedly opened by piercing with a needle. Thimerosal is one common preservative that's been kind of phased out of usage in favor of others, but this was a target of anti-vaccine controversy because thimerosal contains about 50% ethyl mercury by volume. Notably, this is a different form of mercury than the type that actually causes mercury poisoning, which removes that concern, and the amount of mercury that's introduced by thimerosal is also lower than trace amounts that someone would ingest by eating certain types of fish, so preservatives are not really a safety concern. Vaccines also require stabilizers like sugar or forms of gelatin that can help keep the vaccine doses stable under controlled storage conditions. These are naturally occurring compounds in the body with no known toxicity effects. Lastly, vaccines may include trace amounts of compounds left over from the industrial vaccine manufacturing process. This includes residual egg proteins that we referred to earlier, which could potentially trigger egg allergies. Formaldehyde is another compound that's popular for citing in misinformation campaigns because this can be found in trace amounts after being used to kill or inactivate either pathogens or bacterial toxins. These trace amounts are about 10 times lower than naturally occurring formaldehyde found within various human tissues, so formaldehyde in vaccines would never be introduced in high enough amounts to cause any type of adverse effects. Lastly, antibiotics are often included in vaccine manufacturing to prevent any sort of bacterial contamination in cell culture or during the protein purification protocol. Because these may be found in trace amounts in the final vaccine product, manufacturers typically use antibiotics like neomycin that aren't usually the target of antibiotic allergies, unlike things like penicillin or sulfa drugs. Going over these vaccine components helps us address any safety concerns about ingredients that are found in vaccines. Importantly, when we look specifically at the most immunologically relevant components, which are the antigen payload and the adjuvants, we can next learn exactly how vaccines confer protective immune memory. Understanding the mechanism of action of vaccines requires us to apply what we've learned so far in this course about different components of an immune response, like we looked at in our last lecture. Our understanding of the mechanisms of immune memory formation allows us to therapeutically manipulate that immune response to our benefit, in this case by stimulating an immune memory response without any of the risks associated with natural infection. Vaccines start with innate immunity, since they deliver a pathogen-associated antigen in combination with inflammatory PAMPs that can stimulate innate immune signaling pathways. For live attenuated vaccines, PAMPs are kind of already encoded within the attenuated viral strain, while for non-live vaccines, adjuvants such as aluminum salts can activate the inflammasome NLRP3. Vaccines are administered in peripheral tissues, often through intramuscular injection. And this is because muscles are well vascularized, which means that it is easy to recruit additional APCs. It's also easier for soluble antigen to drain, which is why intramuscular injection is most often used for vaccines. As we just established, the antigenic component of vaccines can be administered through a variety of different formulations, all of which function along with adjuvants to activate innate immune cells in the muscle. This includes PAMP-PRR signaling associated with APC activation that we learned about in week 2, as well as antigen processing and presentation that we learned about in week 3. The end result of this first stage of an immune response to vaccines is that you get local inflammation and activated APCs such as dendritic cells, and these then display the vaccine's antigenic component on both MHC class 1 or MHC class 2. These activated APCs can then leave the peripheral tissue and migrate to the closest tissue-draining lymph node where they initiate immune priming. At the same time, soluble vaccine antigens can also drain to the lymph node where they bind to cognate B cell receptors, which we will get back to in a little bit. Once an activated DC arrives at the lymph node, it can use the specialized architecture of the lymph node to efficiently browse the T cell zone. This browsing behavior allows the DC to find T cells with cognate TCRs, which recognize the vaccine antigen epitopes that the APC presents on its surface. Vaccine-induced DCs can stimulate both helper CD4 T cells and cytotoxic CD8 T cells, which we learned about during our week 3 lectures on antigen presentation, as well as the signals that are relayed to T cells when they're activated by APCs. We also learned how T cell activation by APCs leads to functional effects of the T cells during our week 4 lectures on T cell biology. In the case of activated CD8s, these can differentiate into short-lived effector cells, and there will also be a subset of activated CD8s that will differentiate towards a memory lineage and form either effector memory, central memory, or tissue-resident memory T cells, all of which can be recalled in response to future exposure to that same pathogen. Importantly, activation of CD4 T cells enables T follicular helper cells to provide T cell help to B cells in the lymph node. And importantly, these are the B cells that have bound soluble vaccine antigens at their BCR. This allows the processes of affinity maturation and isotype switching to occur in germinal centers of the lymph node, which again function by optimizing the antibody response against the vaccine's target antigen. When you give booster shots for vaccines, they typically function by reactivating the germinal center responses against that vaccine's target antigen. Following immunization, B cells can form either memory B cells or long-lived plasma cells that migrate to the bone marrow where they can live long-term and then become reactivated upon re-exposure to that pathogen. So long-term humoral protection from vaccines is a product of both circulating levels of neutralizing antibodies that are maintained after infection, as well as the reactivation of memory B cells that can rapidly produce antibodies. Memory plasma cells in the bone marrow are extremely important for these long-term responses, which is why if you know anybody that's had to have a bone marrow transplant due to something like leukemia, this actually wipes out all of their memory plasma cells that they've had since childhood. So those patients usually then need to get all of their childhood immunizations again after the bone marrow transplant. We learned about the mechanisms of these B-cell responses during week 5 of this course, including the many ways that antibodies offer humoral immune protection. Taken together with what we also know about innate immune activation, antigen presentation, and T-cell responses, this example of an ideal response to vaccination incorporates several of the major topics about the immune system that we've covered so far in applied immunology. Again, vaccination essentially tricks the immune system into thinking that it needs to generate a comprehensive immune response against an active infection, but it does so in a very safe way that maximizes the benefits of long-term immune memory while minimizing disease risk since there's no actual infection. If we understand how the immune system should ideally react to a vaccine in a pretty straightforward way, why doesn't this work for all vaccines that have been developed against all pathogens? The answer, like most things in biology, is that vaccine responses can get quite complicated as a result of numerous biological factors on both the side of the host immune response as well as on the side of the pathogen. On the host side, the age at which you receive an immunization is very important. So older people just don't have immune systems that can respond as robustly to stimulation, so it's difficult to form really good memory populations of B and T cells, which is unfortunate when you consider that the elderly are sometimes more susceptible to serious disease symptoms. And this is especially true following infection with things like influenza or SARS-CoV-2. There are also some pathogens where the endogenous or natural host response to infection just isn't as well characterized. There are also some pathogens that are particularly good at infecting humans, so we can't eliminate them during natural infection. This is an issue because without knowing what natural responses to infection look like, it's difficult to develop a therapeutic strategy of which types of immune responses you want to preferentially target. And this is the case for infections like tuberculosis, malaria, and HIV. There are also considerations on the pathogen side that complicate vaccine design. A major source of this is in high variability pathogens that are constantly mutating and may express modified antigens. So even if protective immunity is achieved against one antigen, new variants could escape. This occurs with RNA viruses since they have higher mutation rates, which is what you see with HIV, influenza, hepatitis C, and potentially SARS-CoV-2. There are also viruses like influenza that circulate as numerous distinct strains endemically within a population. So when designing a vaccine, scientists try to predict which strain will be the dominant one during a given infection season. If that prediction is incorrect, however, then the vaccine may still, technically and biologically work. It just won't work against the viral strain that people are most likely to encounter that season. There are also pathogens with unknown immunodominant epitopes that haven't been characterized yet. This is the case for the causative agents of gonorrhea and syphilis, as well as with the parasite that causes malaria. Without knowing which antigen to target, it's very difficult to design an effective vaccine. Lastly, there are numerous pathogens that simply lack a good preclinical animal model for studying immune responses and therefore conducting vaccine testing. This is an issue with HIV and represents a barrier for advancing our understanding of endogenous immune responses. This chart summarizes several diseases for which there are no effective vaccines, most of which are considerable public health burdens throughout the world, and highlight the need to continue vaccine research in order to successfully develop new preventative therapies. I also want to note that there are several social, political, and intellectual barriers to vaccine efficacy, since these powerful tools only work if they're actually used. Issues related to infrastructure and access are huge problems in many countries, including regions within the US. The prioritization of vaccine research is also important, since unfortunately, a lot of funding for this work is concentrated within relatively rich countries like the US, where things like malaria just aren't really a concern from a very selfish point of view. If there are alternative therapies available for a disease such as HAART for HIV, again, there may not be as much of a drive to develop vaccines if these disease symptoms can be managed. Lastly, the spread of misinformation that leads to vaccine hesitancy is another barrier to efficacy, as we discussed at the beginning of this lecture. I want to end today by talking about an epidemiological principle called herd immunity or herd protection, which is kind of the central goal of population-wide vaccination initiatives. Herd protection is important for protecting members of society who can't be safely vaccinated themselves. In this panel on the left, in a population where everyone is susceptible and nobody has pre-existing immunity to a pathogen, an infected individual, shown in red, can pass infection to susceptible individuals, shown in blue. This is what we observed at the very early stages of the SARS-CoV-2 pandemic. Now, we're currently now in a state that's described by this central panel of the figure, where disease transmission is occurring in a mixed population of susceptible people, shown in blue, and immune individuals, shown in yellow. As the proportion of vaccinated individuals increases, the number of potential new hosts for the virus decreases. So the overall percentage of the population that is still available for infection is effectively decreased. If this progresses far enough, then we reach a state of herd protection that's represented by the right panel here. A high enough percentage of the population here has been vaccinated that infection transmission is significantly interrupted. So even though there may be some individuals who are still susceptible in blue, the statistical likelihood of them actually having contact with an infected individual is extremely low. In this case, a population has reached a state where susceptible individuals are indirectly protected by the high proportion of immunized individuals. It's important to reiterate the fact that vaccines offer us a way to reach herd immunity without losing a considerable percentage of susceptible individuals to death or disability. This is what you would see with the course of natural infection ripping through a population. The percentage of the population that must have immunity to a pathogen in order to qualify for herd immunity depends on the transmissibility of a pathogen, or its infectiousness. For highly transmissible infections, such as pertussis or measles, about 95% of the population must be vaccinated in order to prevent disease outbreaks. For a new infection like SARS-CoV-2, it's difficult to predict exactly what the herd immunity threshold would be, but lower-end estimates are around 70% of the population. As the virus spreads amongst non-vaccinated individuals, we're basically selecting for more transmissible strains, and so you want to note that that could impact the herd immunity threshold. Herd protection is very important for people who cannot safely receive vaccines. And these include babies, immunocompromised individuals, such as certain cancer patients, primary immunodeficiency patients, or transplant recipients, as well as people with severe allergies against vaccine components. The benefits of mass vaccination efforts therefore have an altruistic component, in that they go beyond the direct protection of the individuals that receive a vaccine by extending that protection to members of society who cannot be safely vaccinated. I know that this was a long talk today, but since this is such an important topic that's clearly relevant for our current public health crisis during the pandemic, I wanted to make sure that we dedicated enough time to cover vaccines. I hope today's lecture has helped us establish an appreciation that vaccines are one of the most powerful preventative tools we can use to safely control the spread of communicable diseases. We've summarized the goals of effective vaccines, which first and foremost center on patient safety paired with long-term immune protection. We've learned how different vaccine formulations accomplish these goals, along with other vaccine components. We've also established how vaccines share a general mechanism of action that stimulates long-term antigen-specific immune memory, and how we can take many of the things that we've learned about immune responses in this course to fully understand the biological mechanisms of vaccines. We also touched on factors that interfere with vaccine efficacy for some pathogens that we currently do not have good vaccines for, and how these factors range from biological aspects of host-pathogen interactions to broader social, political, and intellectual barriers. We ended with a discussion of herd protection, or herd immunity, which highlights the benefits of immunization efforts not only directly to the individual, but indirectly to other people who cannot receive vaccines. Lastly, I hope that I've communicated the pressing need to build and maintain public trust and support about vaccines. Given widespread skepticism about vaccines in recent years, effective communication about the underlying biology behind vaccines, as well as their safety, is extremely important. I hope that with what you've learned today in lecture and throughout this course, you feel properly equipped with the knowledge to address these types of concerns, whether they are voiced by patients in a clinical setting or within your own community. Scientific literacy on the topic of vaccines and the ability to communicate this information in a respectful and convincing way is our main hope for trying to realign the public dialogue about vaccines in order to ensure that they are used with high enough rates to maintain their status as a central pillar of public health. That's it for today. Remember that you have an optional Lecture 8C that you can watch this week if you're interested in learning more about the SARS-CoV-2 vaccines that have been recently developed and rolled out. If not, then please still check the final slide of Lecture 8C for reminders about reading, quiz, and discussion assignments for week 8.